My name is Jesse Aaron, and I am an application scientist at Janelia at the Advanced Imaging Center. Oops, get this in presenter mode here. Okay, can everybody see my movies here?
Yeah.
Okay. So I'll start with what I hope is an obvious statement for most of us here, which is that while there are many powerful types of microscopy in biology, fluorescence and electron microscopy are two big mainstays for a large majority of bioscience researchers around the world. And the reasons why, I hope, are illustrated in these movies here. On the one hand, fluorescence microscopy can, of course, give us beautiful images over time and in 3D with this exquisite molecular specificity. But on the other hand, electron microscopy can allow us to drill down into the ultrastructure of biological tissues, also potentially in 3D, as I'm showing here, but usually with much higher resolution than fluorescence. However, while we can see all of these wonderful structures here in great detail in EM, it's generally harder to achieve this really nice molecular specificity that fluorescence microscopy can give us. So it's this particular set of features that I want to highlight today for my talk, and it's this: fluorescence microscopy can take advantage of this huge range of molecular specific labels that are available, to the point that it's possible to label nearly any class of biomolecule pretty efficiently. But in a way, that specificity can sometimes be a weakness, because often what we want to know is the structural context that surrounds a particular biomolecule. In other words, what we often want to know is what compartments are a particular biomolecule sitting in. And to answer that question using solely a fluorescence technique, that would require us to specifically label every single structure that we might think is important, and that gets technically difficult pretty quickly. Electron microscopy, on the other hand, has the exact opposite problem, right? EM images are usually chock full of structural information, and we can generally see all kinds of compartments like the mitochondria or ER and so on, all in a single image. But to know where a particular molecule is within that context is usually a lot harder than it is in light microscopy. So the natural question is, can we marry what I'll call the molecular information of LM with the structural information of EM? And of course, the answer is to perform correlative light and electron microscopy, or CLEM. So to give you a little bit of a more concrete example of what I'm talking about, here is an image from a paper that I'll come back to again. On the left is a fluorescence image of histone 2b that's sitting in the nucleus of a C. elegans cell. And here is the same nucleus imaged in electron microscopy. Now, both of these images are information rich. The fluorescence can show us, the distribution of the histones and so forth. And the EM shows us exactly, for example, where the nuclear membrane is or where the nucleolus are. But I think it's when these two images are combined is where I think things get really interesting, because now we have the capability to say, okay, where are the histones in relation to the different structures in the nucleus? So I hope that you can appreciate that answering these types of questions is rather difficult using either fluorescence microscopy or electron microscopy by themselves. And it's really when you get to the combination of the two that you can start asking some different kinds of questions. But as you might be able to imagine, combining fluorescence and electron microscopy has more than its fair share of technical challenges. And they're all interrelated to some degree or another, but I'll try to classify them into three main groups. The first has to do with the fact that, as I've mentioned briefly, that LM and EM typically operate in quite different resolution regimes. And that can place, at least historically, has placed limits on what we can infer from CLEM images. The second challenge deals with how do we combine these two very often different kinds of data sets in a quantitative way? In other words, how do we accurately register light and electron images with each other to be able to make some meaningful insights or conclusions? And finally, how do we reconcile the fact that sample preparation techniques and imaging workflows are actually quite different and potentially even incompatible between light and electron microscopies? And are there some workflows that can address these issues? So I'll start with our first challenge, and that's the mismatch between the resolution between LM and EM. So I'll start here by trying to illustrate the magnitude of the problem. I think people can understand that the resolution difference exists, but I want to show the exact magnitude of this. So hopefully, as we should all know, any microscope, the resolution is at least in part determined by the wavelength of whatever we're using to illuminate the sample. So if we're using visible light photons, our wavelength is around 500 nanometers. So let's say that our resolution is roughly half that at best. And this blue circle on the left represents that. But an electron, on the other hand, has an extremely small wavelength, depending on how fast it's traveling, but it can easily be in the angstrom range. Practically speaking, resolution of, let's say, 2.5 nanometers in electron microscopy is not unreasonable, which is represented by this small blue dot here. And if you're having trouble seeing that, that's because we can fit 10,000 of these these little blue dots inside this big blue dot. So that gives you, I hope, a sense of the difference in resolution between these two imaging modalities. So I'll show kind of the same information here in this next slide in the form of a graph that I think shows the difference not only in the resolution between, let's say, typical wide field and TEM, but also the difference in the field of view that are usually acquired using these two techniques. But how recently things have started to change in our favor, and that has to do with the development of super resolution microscopy. So I'm going to throw this up here. So while confocal can certainly get us a little bit better resolution, particularly in the Z direction, it's not really until you get to the development of structured illumination microscopy and true super resolution microscopy that we see that the resolution gap between LM and EM has started to narrow. And in parallel with that, there have been some really exciting developments in electron microscopy that have pushed the boundaries in terms of field of view or volume in the case of 3D imaging, that also kind of get the two imaging modalities closer together. And I'm going to highlight some of these developments later in my talk. But before I do that, I want to briefly review some common enhanced or super resolution techniques out there. I know most people are familiar with this, but I just want to go through it quickly. As I said, these techniques can get us closer to the same resolution that we get in EM. But each of these are capable of both 2D and 3D implementations. But the strengths and weaknesses should be weighed carefully when we're thinking about CLEM. On the left here is structured illumination microscopy. In terms of resolution, SIM can offer us a moderate gain in resolution over conventional imaging. Resolutions of maybe 100 nanometers are possible here, but SIM also doesn't require generally particularly high illumination power, at least compared to the others. And I think most importantly, there's really a broad choice of fluorophores that are compatible with structured illumination. When we get to STED, stimulated emission depletion microscopy, the resolutions can usually expect to be a little bit better, but that comes at the cost of both harsher illumination and also at least a moderate degree of flexible fluorophore choice. But on the right here, the single molecule localization techniques like PALM and STORM, they do generally give the highest resolution of these three techniques, but the trade-offs are pretty severe. First, it's not particularly live cell compatible, which may or may not be a concern for us in CLEM, but more significantly, the range of fluorophores that are compatible with this technique are relatively limited, okay? So second here. So I want to go through and highlight a few studies that demonstrated the possibility of super resolution CLEM in a few different implementations. And the first one here came out of Eric Jorgensen's group in Utah. In this case, they had some C. elegans expressing either histone 2B or TOM20 fluorescent proteins. And they went and processed these samples for EM. So they fixed and embedded and made some thin sections. And from there, they imaged these sections using either STED or PALM in each case. And then the sections were imaged in scanning electron microscopy. And they used some fluorescent beads to register the images with each other. And what I thought was nice about this study was that they showed the resolution enhancement between conventional diffraction limited imaging and super resolution. And as you can see with both techniques, but especially with PALM, the resolution enhancement is pretty significant. And I think it really lets us be able to more meaningfully compare the light and the electron microscopy images together. And here is another example from Marcus Sauer's group in Germany, where they combined storm imaging, again, with scanning electron microscopy, but this time of nuclear pore complexes. But instead of sectioning their samples in the microtome, they were able to just isolate the nuclear membrane and lay it on a flat surface. And then they chemically fixed it and labeled a couple of different components of the nuclear pore complex. First here, they labeled the core, and then they labeled, I believe it was GP210, which is localized to the periphery of the NPC. And so first they performed storm imaging on these samples, and then they processed their samples for EM and imaged the same region of interest. And interestingly, they were able to register the LM and the EM images using the nuclear pore complexes themselves, rather than using some sort of external fiducial, as was the case in the previous slide. So what did this give them? Well, you can see the SEM helped them identify some nuclear pore complexes that were not labeled, which was sort of puzzling. But also the storm imaging indicated that some nuclear pore complexes didn't display the canonical 8-fold symmetry, but rather a 9-fold symmetry. So I thought it would have been interesting to see if there were any morphological differences that predicted this in the EM image. But in any case, what you'll notice in this example and in the previous one, all the images are in 2D. So we don't have a Z dimension anywhere. So that brings me to this next example. And this came out of Harold Hess's lab some years ago here at Janelia, where they combined interferometric PALM or IPALM with focused ion beam scanning electron microscopy or FIB-SEM. Both of these imaging modalities are 3D in nature. So what they were able to do was to label cells for TFAM, which is a mitochondrial DNA binding protein. And then they fixed and sectioned the cells into relatively thick sections, like I think it was about 750 nanometers. And they performed IPALM imaging on the labeled TFAM, and then they processed the same sample for FIB-SEM. And I'll talk more about FIB-SEM later and some of its advantages later on in this talk. But from a biological perspective, this allowed them to view the positions, for example, of the mitochondrial nucleoids relative to the cristae. And this allowed them to observe some different morphologies here. For example, in some cases, they saw the nucleoids occupy sort of this empty space inside of the mito away from the cristae. In other instances, it appeared like the nuclei and the cristae were sort of intertwined with each other. But it was really this 3D capability that I think shows a good example of what is possible. So I'll talk more about 3D CLEM here shortly. But I want to come back to our 3 interconnected challenges and now talk about registration. So when we talk about registration, and obviously the main goal of CLEM is to be able to correlate the light and the EM data together, we need to think about 2 things on 2 levels. First, we want to talk about at the data acquisition stage, where we merely want to locate the corresponding regions of interest when we transfer our sample from the light microscope to the electron microscope. Otherwise, obviously, there's nothing to correlate, right? The second level comes after data acquisition, and that's when we have 2 image sets of roughly the same field of view. How do we align those 2 data sets in the most accurate way possible so that we can start making some meaningful insights and generating results? So first, I'll talk about this first stage here. So when we're talking about locating the same fields of view between 2 different imaging modalities, there's at least two common ways. And the most common way is to use what are called locator or gridded cover slips. And there are equivalent versions if we're using TEM grids instead. But the concept is pretty straightforward. These substrates will typically have a pattern or a grid etched into them, usually with some kind of letter and number coordinate system. And that allows you to locate a relatively small field of view onto your cover slip. And this pattern should be visible in the EM as well, either if you're using SEM, just using that same cover slip, or if you've embedded your samples in resin, the pattern can be transferred onto the surface of the resin block too. So that allows you to locate roughly the same region of interest in both imaging modalities. An alternative way is you can actually use just the unique patterns in your sample combined with either low magnification imaging or tiled imaging of your whole sample to find your corresponding regions of interest. And here's an example of something that I did where we labeled some neurons on a 3mm cover slip, and we took a montage image to basically get the location of all the cells on our sample. And then after EM preparation, we took an image of that same sample, in this case using a micro X-ray CT system, and through some just relatively straightforward manipulations just in Photoshop, we were actually able to overlay the fluorescence image just by using the unique distribution of cells in the sample itself. And that allows us to find where our cells of interest are within the resin block itself. So this is an alternative method. But it's probably the gridded cover slip method that is the most common, and those are available widely from multiple vendors. Okay. So now that we've found the same region of interest in both the light and the electron microscope, and we've acquired our images, we need to perform the final image registration. And at its heart, registration depends on finding some kind of fiducial markers that are visible in both image sets. And historically, the most common markers have actually been externally added markers, that is, materials that are added to the sample. And these can include things like gold nanoparticles or quantum dots or fluorescent polymer spheres. But the main requirement is that, of course, they need to be visible in both imaging modalities, but also they should be well distributed in space in the sample. And I'll get to the reason why here in a minute. But I'll add one thing, which is, in theory, this is equally applicable to both 2D and 3D data sets. But if you can imagine, if all of your fiducials are adhered to the surface of a cover slip or a TEM grid, the alignment in that third dimension is going to be harder, right? Because all of your fiducials are in a single Z position. So that could kind of potentially violate that second requirement, that they're distributed well in space. So that brings me to another idea, which is, you can use biological structures themselves to act as fiducials. And I'll talk a little bit more about this in a minute, but most biological structures are distributed in 3 dimensions. So this can make the 3D registration easier. But again, the images need to be apparent in both image types, and they need to be well distributed. So here's an example from a paper that came out at Janelia that I'll get to more later. But the idea was that they found that the mitochondria, and to some extent the endoplasmic reticulum, if you fluorescently tag them, can act as sort of these internal biological fiducials. So once we have these fiducials or waymarks identified in both of our image types, we typically need to digitally transform one image so that it is registered as best as we can get it with the other. So say we have 2 images here and we have identified the fiducials in both of them. What do? Well, the simplest case is what we call rigid alignment. And that typically means that all we can do to one image is to either rotate the image or maybe scale it to make it bigger or smaller. And then, of course, you're going to probably have to shift the image to get them registered with each other. And in some instances, that's sufficient. That example where I showed from Marcus Sauer's group, where they were imaging nuclear pore complexes, they used just this kind of type of transformation. But very often, we end up with something that looks like this after a rigid alignment. And that's when we need to turn to what's called nonlinear transformation, where we actually have to locally warp one image so that it matches the other to a better degree. And the reason why is that, through all the sample manipulations that are needed for EM, the fixation, the dehydration and all the embedding step can actually physically warp the sample. So Computationally, this can get fairly complex and labor intensive to really align light and EM data sets, particularly large 3D data sets with good accuracy. So fortunately, there are a number of software tools out there that can help you on your way. And a number of them are open source, fortunately. In terms of capabilities, they can range fairly broadly. Some can do only 2D data sets. Some can do both 2D and 3D. Some can only do rigid alignment. Some can do non-rigid. To me, a surprising minority of packages out there will actually automatically spit out the alignment error associated with your data. But a number of these packages do offer some automated or semi-automated workflows that can help you reduce the labor time associated with this process. So I just put this table up here to really give a sense of the issues and the capabilities that you need to think about when you're thinking about a CLEM workflow. So that brings me to my third and final challenge of CLEM, and this is really the main task when we talk about CLEM, and that's deciding on what's the optimal sample prep and imaging workflow. So I'm going to spend the rest of my talk discussing this issue and giving a few representative examples that are out there, and then I'm going to focus on a particular CLEM workflow that was developed here at Janelia, and one that's actually freely accessible to researchers throughout the world through the Advanced Imaging Center. So if you've ever even skimmed the literature to kind of get an idea of what a typical CLEM workflow looks like, you've probably been a bit dismayed to learn that there really isn't such a thing as a typical CLEM protocol. There are numerous CLEM protocols out there. Almost everyone varies at least slightly from the others. So choosing or developing a workflow for CLEM experiments can be pretty daunting, I think, especially if you haven't done it before. And you know, the first and foremost thing that you have to think about is the biological questions that you're trying to address first and foremost, but then you also have to think about what are the equipment and the instrumentation that you have access to. And even more importantly, I would say, is the expertise that you have access to. I think you can appreciate that CLEM experiments are technically challenging, and doing them are it's generally going to require some specialized skill that are typically found in core facilities. So that's going to almost always be your first stop when you're thinking about starting a CLEM project, is to go talk to some core facility folks. But that being said, I will try to generalize here a little bit. And one way we can organize CLEM protocols here is based on their order of operations. Most, but certainly not all, can be classified into these workflows where the sample preparation for EM either comes before or after the light microscopy stage. Most of the time you'll see that the electron microscopy happens last, but the first two steps can vary a bit. And what is generally the case in either scenario is that you're going to start with a fixed sample. And that's just because it's a pretty complex task to take live cell fluorescence images and correlate those with a static EM image. And we've already talked a bit about some of the super resolution light microscopy techniques out there. So I want to talk, at least in a general way, about both sample preparation and some of the choices that are available for electron microscopy. So as I said, having fixed samples is usually a prerequisite for doing CLEM, and there's essentially two main ways that samples can be fixed, although I'm simplifying here a little bit because they can actually be combined. But in general, samples can be chemically fixed using one or more of these classes of chemicals. We can use aldehydes of various sorts or alcohols, like ethanol and methanol. And actually the heavy metal stains that provide contrast for EM also act as chemical fixatives. Generally speaking, these kinds of techniques are easier technically, but they can also quench the fluorophores in your sample, and I'll show you an example of that later. Also, chemical fixation is not always going to be great at preserving particularly delicate structures, at least some of the time. So an alternative is to do cryofixation, and here the idea is to literally freeze your sample fast enough that the ice that is formed is not crystalline, it's vitreous. And that avoids that exploding soda can in the freezer effect that would otherwise break the cells apart in your specimen. And there's three common ways to do that I've listed here. There's plunge freezing and slam freezing and high pressure freezing. Cryopreservation is, in contrast to chemical fixation, is usually more technically challenging, particularly the high pressure freezing, which usually requires some pretty specialized equipment. But there are many people out there that say HPF is really the method of choice for preserving ultrastructure. I won't wade into that debate too much, but I will come back to this a little bit in a minute. And just to continue here a little bit, once we have our samples fixed, we can, like I said, either go and image them in the light microscope, or we can proceed through the rest of the sample prep process. And I won't go into great detail here, but we'll perform some steps that typically introduce some heavy metal stains into the sample to provide contrasts, and we'll need to somehow remove water from the sample, either through some dehydration steps, or in the case of cryofixation, we'll do what's called free substitution, where we replace that vitreous ice with some solvent like acetone or something like that. And then finally, the sample will either need to be embedded in resin, especially if it's going to be sectioned, or it can just be coated with some electrical conducting material like carbon for SEM. Okay, so once we have our sample ready for EM, we of course have many, many techniques to choose from, and I'm not going to go through every single one, but I will say if we're just interested in imaging in 2D, of course, TEM and SEM are the most common techniques there, but of course, there's lots of variations within that. For obtaining 3D EM data sets, there are also a number of techniques out there. Electron tomography is a common one, where we just tilt the sample through various angles. But that's usually pretty limited in the amount of depth information that you can acquire. There's also serial section TEM, where we simply take successive sections from our sample and image these in TEM to build up a 3D image. That's usually pretty technically demanding and pretty labor intensive. Serial block face SEM actually involves physically trimming a slice from the top of the 3D block and then doing SEM and repeating this process over and over to build up a 3D image. And finally, there's a technique that I'll talk more about here called focused ion beam. Similar to serial block face, we're sectioning the sample in the microscope, but in this case, we're using a focused beam of heavy metal ions, usually gallium, and that actually ablates the surface of your block, and then we perform SEM over many, many cycles. FIB-SEM is particularly powerful because it allows us to acquire data with isotropic resolution. In other words, the resolution is the same in all 3 dimensions. So I hope the last few slides have given you an idea of really just the vast amounts of choices that are available at each step along the way in designing a CLEM experiment. So I want to go back to a couple of examples that we looked at before, just very briefly, and talk a bit more about the workflows involved in each of them. So if you'll remember this example from Eric Jorgensen's lab, where they were looking at the C. elegans cells. In this case, they did all of the EM prep before they did the light microscopy prep. So they fixed and stained and embedded and made some thin sections, and then they did polymer stead. And there's some good advantages for doing this. It's technically easier, but they had to make some compromises. One of them was that they really had to work hard to optimize their fixation protocols. And even then, they still lost about 30% of their fluorescence. And they also had to work pretty hard to find a resin that was compatible with the fluorophore, because a lot of resins will quench fluorophores. And so they had to choose one that kind of had not a very optimal adherence to the sample. So that caused them some difficulties in handling their sections. But funny enough, some years later, there was a paper that came out of Janelia that talked about a more fixation and resin embedding resistant fluorescent photoconvertible protein, which probably would have made this study a little bit easier. But I think this study gives a good example of where all the EM sample prep steps are performed before the light microscopy step. This example, which again should also be familiar, is one where they did the light microscopy before they did the EM prep. And this approach was nice because it didn't require some of those optimizations and compromises that I talked about in the previous study. They didn't have to worry as much about fluorophore quenching due to the EM sample prep steps. I'll add a caveat here to say, though, that their biological model really helped them in this regard, since they already had this nice flattened nuclear envelope that they didn't have to section. But this illustrates I think that there are some pretty clear advantages in separating all of the fluorescence microscopy related steps from the electron microscopy related steps, because again, it gives us some nice flexibility. So that brings me to the last section of my talk, where I'm going to discuss a particular CLEM workflow that was developed here at Janelia, which I think does a good job of combining a lot of advantages that people are looking for in CLEM. So I'll go through the steps, and then I'll talk about how they can give us some nice benefits. So what we typically do is start with some labeled cells, and then we'll perform high pressure freezing on those samples. And then we go directly to a cryo light microscope. So we're going to image these samples at cryo temperature. And that has some nice advantages that I'll talk about in a minute. But then we'll go ahead and take them out of the cryo light microscope. Then we'll start the EM processing, where we do some freeze substitution and resin embedding and staining and so forth. We'll do a little bit of trimming of our sample, and then we can put it into the FIB-SEM system here. So I'll dive in a little bit deeper here and talk about really the two critical pieces of equipment here. One is the cryo-structured illumination microscope, and one is the FIB-SEM microscope. So this is our CryoSIM scope here at Janelia. It's custom built, as you can see, but it's actually not that complex of a piece of equipment. We do have these really nice sample holders here that can hold up to 5 3mm cover slips, and we take those samples from just from liquid nitrogen and put them into a cryostat. And on the far side of the cryostat is a window that an objective lens can look through, and we bring in the excitation here from the other side. Now, there's some nice advantages to imaging your samples at cryogenic temperatures, it turns out. And the main thing is that we can largely avoid photobleaching in these conditions. It's very, very minimal, which is especially nice for doing 3D SIM, because we can image through the whole cell thickness without really having any signal drop, at the end of our Z-stack. The downside is that we can't use an immersion objective here. We're stuck with air immersion to maintain temperature in the cryostat. So that means we don't have as high of a numerical aperture as we would otherwise. And that's why the resolution of the system is a little bit worse than what you would typically see using an oil immersion high NA lens. But nonetheless, I think we can get some very nice images that I'll show you here in a minute. But the second piece of equipment that I want to talk about is the FIB-SEM systems that we have here at Janelia. These are also customized, but they're based off of existing Zeiss systems. But one of the critical modifications that was made from the commercial FIB-SEM instrument is the system that I'm showing here, that it establishes a feedback control of the focused ion beam milling of the sample. This system actually allows us to measure the amount of ions that are scattering off the sample, as well as the amount that aren't, and also the amount of current that the sample is experiencing. And all of this information can be used to very carefully control the amount of FIB exposure that the sample sees. And that allows us to really precisely control the thickness of the sample that is milled after each imaging cycle. So this, along with several other controls that monitor things like image quality and the environmental conditions, all of these things allow the system to run stably over long periods of time. And that can actually allow the system to shut down very gracefully if there's a problem detected, and then start up again when the problem has been fixed. So altogether, we can acquire data over weeks or even months. That allows us to acquire really large EM volumes. We can get tens of microns in all 3 dimensions, all at 4 nanometer resolution, again, isotropic resolution. So it's a very powerful technology that came out of Harold Hess's lab. So I'll show some examples of what some folks have been able to do with this workflow. So I'll start here with this paper that came out late last year in Nature. And this movie here first shows a 3D FIB-SEM data set of a HeLa cell. They're just showing it in orthoslices here. And then overlaid on top of this is a cryo-LM image of some mitochondria in blue and ER in magenta. And as we peel back and zoom in somewhere here near the midplane, we can see the mitos in the EM image pretty clearly. And these are what they used for the fiducial markers to align the light image. And then we can go back and, for example, add the ER image on top of that. And what's interesting is that they can actually use the LM data to help guide segmentation of the ER from the electron microscopy data. So this is a very interesting synergy that CLEM can give you. It can help you guide EM segmentation, which can be quite challenging. So I think that last slide shows a good example of kind of what's technically feasible. This next movie that I'm going to show you, I think, gives a good idea of some of the biological insights that can be gleaned from this type of data. So here I'm showing a 3D CryoSIM image of a cell that's been labeled with fluorescent transferrin, and that will mark the endolysosomal system of the cell. And then as we overlay the 3D FIB-SEM data of the same cell and kind of zoom in here and then peel back, we can see where the transferrin is in relation to all of the structures within inside the cell. And if we zoom in on this particular spot here, we can peel through the data here and we can see lots of interesting looking vesicles within the milieu all over the place here, in addition to the mitos and ribosomes and all these kinds of structures. But when we look at it in the context of the fluorescence image, that allows us to pick out these endolysosomal vesicles. And using that as a guide, they can then go through and segment these particular vesicles out from the rest of the data. And they saw some really weird and interesting morphologies here, like with this vesiculating tubules and other sort of very interesting morphologies that at least I don't typically associate with endolysosomal particles, which you can see when we overlay the fluorescence image, they were all transferrin positive. So you can say with pretty good certainty what these were molecularly. And then I'll show one more movie here. You know, this workflow isn't just useful from like an intracellular point of view. It can also be used for studies related to cell-cell junctions, so in intercellular kinds of questions. So here's a movie that first shows a 3D rendering of FIB-SEM data taken of some cerebral granular neurons. And what we can do is, if I can get this to play, so what we can do is peel back the EM data to show some CryoSIM data where in green, they labeled a tight junction protein called JAM-C. And then if we bring the EM picture back, you can see a very interesting correspondence between the electron dense regions of the cell-cell junction and the presence of this tight junction protein. And to just kind of hammer home the point here, if we go back and turn the image on its side here, they went through and segmented out the cell-cell junction into high electron density and low electron density regions. So the high electron density is in blue, the low electron density is in red. And again, if we overlay that CryoSIM image on top of that, you can see that this tight junction protein sort of forms this web-like structure in exactly the same places where we see high electron density. So I hope this gives some appreciation of the power of this whole cell CLEM approach, and I hope for CLEM in general. I can also appreciate the fact that many folks out there at the end of the day may be limited by access to some of these technologies and other resources that are needed to perform this kind of work. So that's where my next slide comes in, and that's where the Advanced Imaging Center comes in. So we are an open access imaging facility where scientists worldwide can apply to use any of these pre-commercial imaging systems at the center at no cost. They're freely available. You just have to get yourself here. So in addition to all of these other powerful imaging systems, we are now offering this whole cell 3D CLEM capabilities that I just finished showing you, and that's here. So please feel free to check us out. Go to our website or just shoot us an email. We're always happy to chat with people about their science and how we can help. And you can follow us on Twitter as well. So thank you guys very much for taking time out of your day to listen. And I'll leave it there.
Right. Thank you very much, Jesse. That was a great talk and as usual, packed with information. Yeah. Right, so now typically we're going into the quiz, so I'll just get that up. But I put the link into the chat box, so if you just click on that, you should be able to get into it. And I'll see if I can share that now. Oh, here we go. Yeah, we've got a few people joining us. We are. Let's see if I can. Okay. Right, 7 people. Booyah. All like your talk. Both. Yeah, I think that is the message here. If you can use both. I mean, you know, there's obviously other niches where you need to do live stuff, and then I think we're still away from EM at that point. But you want to see the details, you don't have a great deal of choice. All right, so we've got 8 people. I think we've got enough to get going. So with that, let's kick off. So remember, you've got to be quick. If you want to get that fold scope.
Oh, I didn't realize there were prizes.
Oh yeah, there's a prize. You get to win the great fold scope.
Those are pretty cool.
Winging its way to you. Right, let's enter and the first question. So the faster you are, the more points you get. Which of the following techniques are 3D electron microscopy modalities? That's SEM, FIB-SEM, serial section TEM, or B and C. So it's A, B, C, or B and C is the last option now. Time's up. Oh. 4 people said B and C, so there's 4 people in contention here. Everybody else is going to have to catch up a bit. Let's see if we can catch up on the following questions. We've got 5 questions today. Let's see what's on the leaderboard. The MRC is our current leader with Valeria and Sakura on your tail. So let's see. Next question. And, oh, come on. There we go. What is the typical XY resolution achievable from structured illumination microscopy? Is it 10 nanometers, about 100 nanometers, 300 nanometers, or 1 micron? What's the sort of resolution that you could expect from SIM microscopy? 9 people have gone for 100 nanometers in the right answer, so well done those. Let's see where we are on the leaderboard now. Oh, I think Bongo's bingo might have been faster with Valeria. Yeah, we've got a new winner here. Valeria is winning. Right. Next question. As fast as possible, what is the main source of contrast in FIB-SEM imaging? Is it carbon atoms, fluorophores, heavy metal atoms, or none of the above? And the answer is just coming up. So I had to ask Jesse for the answer. I know, yeah, heavy metal atoms. Okay, a lot of people know more about EM than I do. That's great. Let's see what the leaderboard has to say. Ooh. The MRC is staging a comeback. Bongo's bingo is still pretty quick. Let's see where we go. Valeria is still holding on, but Sakura is just on the tail. So get cracking on this. Question 4. Cryofluorescence imaging provides which of the following advantages relative to room temperature microscopy? Higher theoretical resolution, less photobleaching, lower optical aberrations, or none of the above? What's the advantage of cryofluorescence? Oh, everyone got it. Less photobleaching. Right, let's see where the leaderboard is, because I think now it's just about time, how quick you are on the button. Ooh, neck and neck there for a lot of people. Oh, Sakura, you just squeezed in by 3 points. Right, so it's all on the last question, I think. That is very close. Come on. We got the dreaded lag.
Oh,
here we go. Question 5. Last question. Because of deformations in the sample due to fixation, which of the following transformations are needed to register EM and LM data sets? Translation, scaling, rotation, nonlinear warping, or all of the above? Right, I think it's whoever's fastest is going to get this one. All of the above is the correct answer. Right, let's see where we are on the leaderboard now. Oh, bingo, I love that name. Sakura, you held on to your lead, so well done. If you could just put a contact detail into the chat, we'll send you a fold scope, and thank you very much for everybody for participating. That was quite good fun. All right, and also for everybody else, put your questions into the chat or stick up your hand and you can actually ask your questions live. All right.
Actually, Nick, I will have a go. I have a question. So Jesse, you indicated this nice paper from Marcus Sauer lab where you observed like 9-fold symmetry of nuclear pores in addition to it. I almost felt you did some experiments to disentangle those. So actually, a comment I have is 9-fold symmetry is not very new. Like Joe Gall, who actually discovered nuclear pores, he works not too far from your place at Johns Hopkins campus, Carnegie Institute of Washington. So he found like a lot of nuclear pores had 9-fold symmetry. I think the issue is there is another paper from Jonas Rees' lab where he establishes nuclear pores as the reference, where he has the 8-fold symmetry model, and whether you have 5 components to the pore subunit, 6, 7, 8, 9, you just average them to the 8-fold symmetry model. So I think that's one of the issues, how the standardization has been pushed. So everyone looks for 8-fold. But nonetheless, I'm curious if you did some experiments to address this, and how would you handle it? Like, do you think like there is let's say we have 100 nuclear pores in the field of view, a population of them are 9-fold, not significant enough, maybe 10 of them. So how would you approach this problem, and how would you complement? Did you do any EM experiments to complement both?
Yeah. I'll try to talk generally here. I mean, first of all, I'm not a nuclear pore complex expert by any means, but I think this 8-fold versus 9-fold symmetry shows first of all, the power of imaging, right? We can, instead of getting a global average, we can get, individual nuclear pores in the field of view. My thinking about this experiment or this paper in general was they had CLEM, right? It would have been cool to see a little bit more investigation to see, is there a correlation between those pores that have an 8-fold versus 9-fold symmetry and some kind of structure that they can discern from the SEM? I think that those kinds of questions are the type of questions that CLEM is good at answering. I think, whether or not this is a canonical structure or not, it's a very good question. And of course, NPCs are really one of these model systems for Palm Storm microscopy, and some of the work that was done in actually making average nuclear pore complex images, I think is extremely powerful, and we've been playing a lot with this too. But you're right, when you have potentially 2 different populations of a structure, what is the average structure? Or if you do try to make an average structure, are you taking some weighted average of multiple populations? So there is I have seen some work out there where you can actually try to separate different structures in a feature space so that you can, if you want to do this sort of multi-particle averaging techniques to really get really high resolution images, that you don't sort of muddle the 2 populations together. But I think it's fertile ground for sure.
I almost felt that you have done an experiment and you know the answer.
Okay. Thank you very much for that. We got a question from Lothar Shermala. Do you want to ask directly, actually, Lothar, since you're still there, or should I just read it out? Where is he?
Yeah.
Here he is.
Lothar. I'm around. Hi, Jesse. Great
Hi,
talk.
how are you?
It was fantastic.
Yeah,
I was wondering about the free substitution step. Is there a danger of introducing any artifacts?
Yes. I think the main artifact that you have to, I don't know if I would call it an artifact, but a challenge that it can introduce is whether or not we do chemical or cryofixation, and most of the time we're going to have to pull water out of the sample, right? And in doing so, this can create this warping, local warping of the sample during process, which is very difficult to predict. And in fact, if you go back and look at, for example, David Hoffman's paper, you can see how certain parts of the cell really don't move much after EM prep, and some parts of the cell really do, and it's hard to predict. So I think it's dehydration, at least in from what I can see, tends to be the step that can cause some warping of the sample that can make interpretation a little bit trickier.
Yeah, in terms of ultrastructural effects, you think it's
it's hard to compare, because I think with chemical fixation you also have similar potential artifacts in preservation. I think because most of the free substitution happens at very, very low temperature, you only get to the room temperature at the very end. I would expect it to be less perturbative than for example critical point drying or ethanol dehydration or something like that.
Thank you.
But I can't say that it's artifact free, right? I don't think we have a gold standard to compare it to, but I don't think it's likely.
All right, thank you very much.
Good question. Good question.
Yeah. So, if anyone else got some questions to answer, I have a couple to carry on the cryo theme actually. So, what is the actual NA that you're getting on your CryoSIM, and what do you think the limit is there?
Well, the limit is the fact that we have to use air objectives, because if you can imagine, if we have any sort of immersion, it's going to start. You're going to start getting heat into your cryostat. So that's the ultimate limit. The NA that we have on that system, we have a 100x Nikon 0.85 objective on there.
Okay.
So I think maybe there's 1 or 2 0.9 objectives, but we also have to have a fairly long working distance, because we have to image through a fairly long distance to see our samples. So it's a challenge. It's a definite limitation for sure.
Right. And just to quickly follow up before we go on to the next question. So is your objective at room temperature then?
Yes. So
Yeah.
all of the optics up to the objective lens are at room temperature, and it's just the sample that's at cryo.
Right.
And the internals.
Super. So we have a question from Abigail. Are you asking question in person there?
Sure. Hi.
Hi.
Here we go.
Jesse, great talk.
Thank you much.
Really wonderful. I run a core facility at MIT, and your talk basically just described our daily existence. Really nice to see. I want to steal your slides so I could present these to all of the people that don't know anything about this. It would save me a lot of time.
Feel free. I'm happy to share anything.
We do a lot of the CryoCLEM and just room temperature CLEM stuff that you were talking about. It's so exciting to see this. I'm wondering, and this sort of gets a little bit to what Luther was asking about the stretching and changes during dehydration and free substitution and possible artifacts. And you have the Zeiss SEM FIBs there that have beautiful contrast even without the metal staining during free sub. And so I'm wondering if do you ever take some of your cryofluorescent samples and go towards just a straight up CryoSIM FIB milling workflow?
We haven't done cryo FIB-SEM simply because we don't have a cryo FIB-SEM. We do have cryo TEM here, and yes, I know that David Hoffman especially had done quite a bit of this, where he did CryoSIM and then gave the folks in the cryo EM facility some of his samples. And so yes, that has been done. I haven't done it myself, but it is absolutely possible. Yeah, and that way the whole thing stays at cryo.
Yeah,
it's very cool. Very cool. I would love to get a cryo FIB-SEM.
Come visit us. It's amazing.
Okay. If I could trade you just one of our FIB-SEMs for one of your CryoSIM, well, let's make it happen.
I saw your lineup of like 5 FIBs. That was impressive.
There's actually 9 now, believe it or not.
Well, we'll take your cryofluorescent, your SIMs, because we don't have that higher res for our cryofluorescents. And as you mentioned, the room temperature or air objectives.
Yes, it's a limitation. I would very much like to also try some cryosted, because one of the limitations of SIM is that we can't really do super deep, super thick samples. And so trying a cryosted, which has been done, and in fact, there's some similar to the CryoSIM, there's some nice photophysical advantages of doing STED at cryo temperatures. You can get better depletion with lower laser powers and things like that. So ostensibly, you can get a little bit higher resolution at cryo than you can at room temperature with STED. So that would be something I would love to try.
That's one to look out for in the future then.
Yes.
Do we have any more questions coming in? Oh, someone's just posted to say that Janelia has an Aquilos cryo FIB-SEM.
We have a Krios, yeah, that might be an autocorrect.
Okay. Well, I guess if we don't have any more questions, I think it just remains to say to thank Jesse for such a wonderful talk. The slides will get posted on the RMS website later on, so you can go and find them there. I think it'll end up on the YouTube channel. Other than that, I think I'll just give you the usual awkward Zoom clap for giving such a great talk, and thank you.
My pleasure, my pleasure.
For getting up and giving us a talk this morning. Okay, with that, I guess we are done for the day, and thank you. All right.
Thank you, everybody.
Okay, Jess, if you're there, you can stop recording.